it is present or expressed on the surface of three professional cells known as antigen professional cells: dendritic cells, B cells, and macrophages. However, dendritic cell is the most professional antigen presenting cell, as this major histocompatibility class 2 is constitutively expressed at high level on the surface of dendritic cell. However, in case of macrophage and in case of B cells, they are expressed at high level following activation. This major histocompatibility class 2 is formed of two chains, alpha chain and beta chain. Alpha 1 consists of two domains. Alpha chain consists of two domains: alpha 1 domain and alpha 2 domain. Beta chain consists also of two domains, beta 1 and beta 2 domains, and they are anchored to the surface, like in major also histocompatibility is anchored to the surface of nucleated cell, and here they are anchored to the surface of the antigen presenting cell. And usually they present peptides that accommodate a peptide, or that they lie here or sit here within this cleft. It is known as peptide binding cleft between alpha 1 and beta 1. It is of around 13 to 18 amino acids. These molecules, or these two chains of the major histocompatibility class 2, they are encoded by genes located within or in the short arm of chromosome number 6 in human, and these genes are HLA-DB-A1, HLA-DB-B1. However, they can be also encoded by other set of genes known as HLA-DQ-A, HLA-DQ-B1, and HLA-DR-A1, HLA-DR-B1, B2, B3. So HLA-DR is highly polymorphic. And these antigen presenting cells, they express major histocompatibility class 2 in association with a peptide within the binding cleft and presented to T helper cell. Then T helper cell, through its receptor, interact with the peptide within the binding cleft, and a protein on the surface of T helper known as CD4 interact with the B domain, beta 2 domain of the beta chain of the major histocompatibility class 2, and this result in activation of the T helper cells. A third class of major histocompatibility class 3 is present in between the loci that encode the major histocompatibility class 1 and the genes that encode the major histocompatibility class 2, and they are present here, but however they express this area of genes, they express soluble materials like some complement, for example C4 and toxic shock protein and tumor necrosis factor alpha. So the major histocompatibility molecules are very important for recognition and discrimination between self and non-self. Thank you very much, Professor Ahmed Boulad.